Well, good morning, everybody. Thanks for joining us. My name is, welcome to the Coffee Seminar Series. My name is Dr. Carl Robinson. I'm a UQ Amplify researcher at within Coffee, the Centre for Horticultural Sciences, and I've been given the honour of introducing your Coffee Seminar Series speaker today, and that's Dr. Alice Hayward. But first, we'll do our acknowledgement of country, and UQ acknowledges the traditional owners and their custodianship over the lands on which we meet, and we all pay our respects to their ancestors and their descendants who continue the cultural and spiritual connections to country, and we recognise their valuable contributions to Australian and global society. And there's a few housekeeping things to mention with the seminar series today. We're scheduled to go from 12 till 1. And at the conclusion of the seminar, we'll hold a Q&A session. Now, if you have questions and you'd like them answered, please put them in the Q&A button at the bottom of the screen and not in the chat button, and we'll address them at the conclusion of the seminar. So we'll introduce our speaker today, and this is Dr. Alice Hayward, and she is a UQ Amplify researcher within the Centre for Horticultural Sciences and at Coffee here. She's a plant molecular physiologist in the MITA lab, and her interests are primarily using biotechnology to understand plant form and function and improve sustainability in horticultural industries. She's very passionate about, she's also very passionate about restoring and preventing further degradation of the environment. A little bit about the MITA lab team and its specific focus on genetic and tissue culture technologies to improve the understanding of productivity and develop commercial propagation solutions for crops in close partnership with industry. The team also has a very dedicated research stream in cryobiotechnology, and this is used for the conservation of quite recalcitrant crop germplasms, and as well as rare and threatened plant species. So their aim here is to help our horticultural industries and environment become more resilient to the ongoing issues of climate change, and assist with the conservation and the recovery of endangered plant species through tissue culture innovations. Their flagship of the team is Avocado, which we're going to talk about today. And they've had a number of firsts, this team, including the production of the first genome sequence for avocado, as well as the world's first commercial tissue culture pipelines for avocado propagation and cryopreservation. They've got a pretty significant ongoing vision, and it's the extension, and it's to extend these technologies to new species and crops for both horticultural and environmental outcomes. So with a resume like that, without further delay, I'm pleased to introduce Dr. Hayward's talk, From Lab to Orchard and Beyond.
Thank you, Carl. That was a really lovely introduction. I appreciate that. Just checking everyone can see me, and that's fine. Okay, so yes, thank you. I really appreciate being invited here today to speak on behalf of our team, Avocado Tissue Culture team, in Nina's lab, taking the place of our fearless leader, Nina, here today. And I'd like to acknowledge as well the traditional owners and their custodianship of the lands on which we're meeting today, and pay my respect to their ancestors and descendants. Other people I'd really love to acknowledge before I start is this whole team, our tissue culture team. All the slides have come from our group effort that we presented recently at a growers outreach day, and that I've kind of modified for this talk today. So all the way from Nina through to Maddie and Jini and the whole team, thank you. So today I'm talking about our avocado tissue culture, from lab to orchard and beyond. Okay. Sorry, here we go. So why avocado? I always love to say, you know, it is the most trendiest food, so why not? It's the most pinned food actually in the world. one of the most Instagrammed fruit species you'll ever find. There's memes aplenty about avocado, and it's even made Nina famous here, with a master chef here producing her Indian guacamole on TV there. You can even use avocado to propose to your girlfriend if you like. He didn't choose a very good one, I don't think. It's a bit brown there, but there you go. So I don't know if you guys remember the great avocado shortage of 2017. There's a sign here from our local Graceville supermarket saying no cash or avocados kept on premises overnight. So a few of these signs were popping up everywhere because people were stealing the avocados. They were worth about $8 each at that time. And the reason we love avocados is not just because they taste great, but they're really good for us. So they've got amazing amounts of heart healthy fats that help control blood sugar levels as you're eating them, and packed with vitamins as well, which is rare for a fruit to store its energy in fat. So it's one of those pretty cool species. They're so popular, in fact, the demand for avocados is growing phenomenally. The main producer in the world is Mexico, which is also where all the drug cartels are. So the cartels have actually started taking over avocado farms and stealing the crops, because that's where they're making a huge amount of money. So these are actually farmers, avocado farmers, that are protecting their avocado hoard from the drug cartels over there. If we come back to Australia, which I think is a bit more pleasant perhaps, and much more of a small fry on the world avocado stage, but nevertheless a very important crop for us here, especially in Queensland. You can see that over the last 20 years, the production and value of avocado has really increased quite steadily. We now produce over 87,000 tonnes of avocado a year, and it's worth almost $500 million to the Australian economy. And Queensland is a really important factor in this. So we produce almost half of the avocado crop at the moment, followed by Western Australia. So for Queensland, this is a really important crop horticulturally. The great thing about Australian avocados is we can actually produce them all year round. So we have an expansive climate range and conditions, and 8 growing regions around Australia. Hass is the variety you guys probably see in your local supermarket most of the time. It is 80%, makes up 80% of our Australian and global fruit market, all cloned from an original tree that was found in a backyard in California and has taken over the world. Shepherd is another common one. This is the green skin avocado you'll find in the shops. It makes up about 17% of production, and it's the dominant variety during our summer season. In Australia, we really produce almost all of our avocados for domestic consumption, and we eat them all, except around 5%, which is being exported at the moment to a few countries in Southeast Asia. And there's ongoing efforts to grow this export market for Australian clean and green, healthy avocados. Over the summer season, we actually get imports from New Zealand. This is the only avocado that we import. We don't take any other imports from the major producers in the world. So our industry is kind of protected in that way. This graph just shows you that basically over the last 20 years, both our consumption and basically supply of avocados has doubled. We eat about 4 kilograms each every year. I probably eat more than that. And I'm lucky because I get quite a lot of free ones from the field. So it's really growing. And you can see that even now, the domestic supply isn't able to reach the consumption. And the light green bar here is where the New Zealand imports are coming in to supplement that. So in Nina's lab, we have a team with quite a diverse skill sets, including in the tissue culture team itself. We've been working on lots of things to do with avocado, not just propagation, but a lot of genomics and molecular work, as well as Chris, who does avocado cryopreservation, and he gave a coffee seminar not long ago on that amazing work that he's doing. But today I'm talking about our propagation work. So how is avocado actually grown? It's a grafted crop. So that means you actually have a scion, which is the Hass or the shepherd fruiting variety that you graft onto a rootstock to produce new trees. And these trees are what's used for orchard expansion and new orchard establishment. So you actually get this small piece of a cutting, or what's called a budwood, from fields of Hass trees growing around Australia. And you take this mature material and you graft it to either a seedling rootstock or a clonal rootstock. Seedlings are produced by germinating seed, as you can imagine. Clonal rootstocks have the advantage that they are genetically homogeneous, and they're identical to the tree at which the cutting came from. So they're basically cutting produced rootstocks. And the rootstock has a really important role in avocado productivity, tree architecture, nutrient acquisition, and tolerances to soil-borne diseases and salinity and stresses like this. So here's Janie here actually grafting some avocados in Anderson's nursery down in Jaramba. So why do we produce clonal rootstocks? Basically, they allow you to get a really high-performing rootstock tree that you know might be salinity tolerant or disease phytophthora tolerant, and you can clone it so that you can replicate that across all your rootstocks produced for your field. The problem with avocado is that it's incredibly difficult to make clonal rootstocks traditionally. This is a super complex process that nurseries have been trying to do for many years. It still requires a seedling. So you actually start with this seed here that you germinate, and it grows to be a small seedling. Then you chop off the actual seedling, this random seedling, and you get a piece of a cutting from a mature rootstock tree that you want to clone, and you graft it to this seedling. Then this whole plant has to be put in the dark for a few weeks. So thousands and thousands of plants being put in dark for a few weeks until they start to shoot out, these little etiolated shoots. These little shoots that come off the rootstock cutting are then what you use to root the plant. So you put a little pot around it, you treat it with a rooting hormone, and then you wait for the roots to form. So already you've gone through one grafting stage, a really dark period. You've treated the plant with rooting hormone to get these roots to form. And then once roots are there, this is where you actually chop off your rootstock shoot and you get your Hass or shepherd budwood and you graft on. So this then becomes your grafted plant, and it's still attached to this nurse seedling here. Once they're ready, they can get chopped off the nurse plant. The nurse plant gets discarded, and then you have your clonal rootstocks already grafted to your Hass. In Australia, you might only get one plant, not two like this, and it's really commercially non-viable for a lot of nurseries. It's so labor intensive, it's so time consuming. There's multiple points of loss. You can see every time you graft something, you're going to lose plants. Every time you try root avocado, you're going to get ones that don't root. So it's really, really difficult to clonally produce avocado rootstocks. But it's not just clonal rootstocks that have limitations. So even seedling produced plants, you're limited by the availability of seeds in Australia, which are produced seasonally. So you have a seasonal influence on seed production and rootstock production. They bring pest and disease risks to the nursery. You actually have to maintain huge swathes of land growing avocado trees and fruit just to get rootstocks to make new plants, and that fruit gets discarded. The other problem with seedlings is they're all genetically different to each other. So just like you and me, every avocado seedling is different, which means you can't guarantee the quality of that rootstock. Clonal rootstocks, as I said, can guarantee the quality of the plant, but you still need that really complex process. You still need seed, seasonal seed, you need budwood, super resource intensive, multiple loss points, and you're still getting the plant and disease risks. But clonally produced plants are the only way to guarantee the genetic homogeneity of the root system. And it's the only method you can actually use to propagate improved or rootstocks that are protected by plant breeders' rights. So a lot of the newer rootstocks coming out of nurseries, big nurseries that are creating better selections for improved disease tolerance, these must be clonally propagated. So in Australia, we have this growing demand for fruit. There's a growing Asian market demand where we're situated close enough to hopefully be able to tap into. And we have the climate for year-round supply. And our fruit's good quality, nicely produced fruit. And we have this industry challenge. We can't get plants out quick enough to improve our production or rate of production to growers. And it's not just about supply, it's the desired rootstocks are not available. So it's these plant breeders' rights protected ones, or really highly in demand rootstocks with better traits like phytophthora tolerance, disease tolerance. They're just not available. So there's a time lag in getting rootstocks to growers. There's also a time lag of getting new and improved varieties into Australia and out to growers, because it just takes too long to bulk up the supplies of these clonal rootstocks. And then it creates diseases and biosecurity risks as well. So we bring in our superhero Nina here, tissue culture superhero, with the mission of comprehensive tissue culture propagation for avocado rootstocks to meet industry demand. So if I introduce tissue culture, it's basically the aseptic culture of cells, tissues and organs under controlled environment conditions in vitro. You can start with a really small explant, like this little bud here, and you can basically multiply it to many, many clones in a really happy, controlled way. It's actually a propagation system used already for many, many crops. It's the only way we produce bananas. It's used for papaya, blueberries, potatoes, apples, pears, and others. So it's not new, but it's definitely new for avocado. So when we first started this project on avocado, we thought of what are the benefits for tissue culture production of clonal rootstocks of avocado. It's going to eliminate the dependency on seeds for rootstock production. There's no seasonality to production. We can produce plants all year round using tissue culture. It's independent from climate, weather events, outside stress affecting seed production and budwood production. We reduce the losses, so there's no nurse grafting, no seed germination problems. It's disease-free. Because you're culturing in aseptic conditions, the plants you're producing are disease-free. You can achieve mass propagation of plants from a small amount of starting material. Your plants are going to be true to type, so clonal to the mother plant. And you can scale production depending on your needs, which is really commercially important. Also, as I mentioned, it's essential for those elite rootstocks. So when we first started, if you looked up the avocado literature for tissue culture, people have been trying for a long time because the benefits are clearly valuable. But with over 50 years of research, there was just multiple problems. It's a really difficult species to get to work. Very low multiplication, loss of vigor in culture, couldn't stay in culture for a long time, terrible quality of the plants, extremely difficult to root. So the rooting stage, which is the major bottleneck for the nursery clonal propagation, it's still a problem for tissue culture propagation. These plants don't want to put roots out. Low ability to then acclimatize plants. So once they've come from culture in this perfect environment, they need to make it in the real world. And they often die at that point. So there's so many problems in the literature to date with practicality for large-scale production of avocado. And at the moment, until our research, there was just no commercial tissue culture propagation of avocado in the world. So here comes Nina again, flying in with her tissue culture plants. So just to explain, this has been 8 years of research. This project started with Nina getting an ARC linkage with growers' support, Jasper Farms, Anderson Horticulture, Delroy Orchards, and with Department of Agriculture and Fisheries. And in that project, we recruited a bunch of people, including myself, and these awesome guys here, Chris, Jani, and Maddie. And it was Jani's PhD, actually, that developed the proof of concept in this project for the very first tissue culture system for avocado. She was able to get shoots, and she was able to get roots forming on those plants. And she got her very first tissue culture plants in the nursery and into the field. So this was our proof of concept. We then took all the partners, grower partners from this project, and they joined with us again, and we were successful in getting an Advanced Queensland Innovation Partnership to adapt our system to commercial kind of feasibility. And in this project, we joined hands with 2 other universities, University of Southern Queensland and Central Queensland University. So we did all of the commercial scale up of the technology, got more rootstocks going in culture, established grower field trials, and then USQ and CQ researchers came on board and they helped us with economic analysis and industry survey to really inform our research and uptake of the technology at the end. And this culminated last week in us giving an industry showcase of the work from this project. We also got a bunch more growers on board, and these guys have been really essential to our grower-led field trials. So I'm going to talk about the results from this project now, as this is coming to an end, and I think we're really pretty excited about what we've been able to achieve. So our approach all along, and this is Jaine's brainchild, is getting your tissue culture proof of concept going. You've got to scale it up. So for commercial feasibility, you've got to scale up your experiments from 20 plants to hundreds of plants and make sure that you're still getting the same efficiency and rooting efficiency and multiplication going. You've then got to get those plants acclimatized beautifully, happily growing in the nursery, check that their morphology is fine. So with tissue culture of any plant species, you can introduce somoclonal variations depending on the culture system and how long plants are in culture. So it's important to look at the morphology of plants coming out and just check everything's fine. And then of course our productivity trials and our plant trials in the field. So the way Jaine did this in her PhD was she used a technique called meristem culture. And this is how we've had such good success. This is where you can actually take the extreme apical tissues or a bud with no visible leaf primordia, and you can multiply that up in the lab and get hundreds of plants at the end of it. So in our lab, we start with these clonal mother stock trees, which we maintain in the glass house. We take a little cuttings from these trees with bud nodes in them, and we put them in culture in a sterile way. So we have to sterilize these without killing them and then get them to initiate in culture system. We then teach the little meristems to start to grow and then teach them to multiply into lots of little buds. Then we get these buds to elongate into shoots, and then we can take single shoots and get rooted plants. So Jaine gave a really beautiful presentation at our Growers Day the other day, and she likes to explain this whole method as complexity and comprehension, or teaching languages to multiple different cultures and language groups. So she actually speaks to her plants using all these different factors. So macronutrients, plant hormones, micronutrients, the way she treats the plant, the light and growth and temperature conditions, the pH, the energy source. She modulates these on a really, really fine scale to teach every single plant what she wants it to do. Does she want it to multiply into lots of buds? Does she want it to elongate its shoot? Does she want it to make roots? So she's learnt this language over many years as a tissue culturist, and she can really fine tune how she speaks to every species she works with. And one thing we found with avocado is once you've got one cultivar in culture, you're definitely not set. Every single cultivar has behaved really, really differently. They all have come from different cultural groups. They all speak totally different languages. They don't understand each other. So Jaine had to invent a language for every single one to get them to do what she's asked them to do to make new plants in culture. So we call her the wizard. So reed is a cultivar that's a common Australian rootstock. It's phytophthora susceptible, but it's productive if phytophthora is managed properly. So this was the first plant we actually had in culture from previous work we were doing. And it was also the one that spoke Jaine's language much more easily than the other cultivars. So it was the first one we got working. And this is just showing you in pictures, meristem induction, multiplication, elongation, getting single shoots forming, and then getting rooted shoots from here. So this took many, many thousands and thousands and thousands of different language combinations, but she got this to work for cultivar reed. And with this, she can actually take one meristem from a plant and she can get 500 clones from it. So huge throughput advantage. She then was able to translate this to Velvic. So Velvic is an Australian rootstock that's in really high demand. It's productive and it's got really good phytophthora and salinity tolerance. So this is the one our industry partners really wanted us to get done. And it was also the biggest beast. It didn't speak any languages very well at all. But she managed to get all the way through the system. She's got these beautiful plants coming through now, getting some commercially feasible rooting success now. And she can actually achieve 200 clones of Velvic from a single meristem, which is a super, super advantage for this really popular in-demand rootstock. Zutano is another one we've tried, so I'm just going to go through a few of the rootstocks now. If anyone interested in the ones we've managed to get working, this is another rootstock that's used commonly in Australia and that has passed all the way through the system with good rooting percentages now. Same for Kid, another Australian selection. It's doing well in culture. And then Juicer. So Juicer is one of these elite varieties that's protected by plant breeders' rights. It's in high demand internationally. It's produced by a South African nursery, one of the biggest nurseries in the world. And it has commercial phytophthora resistance and salinity tolerance. So this is one of the globally in demand rootstocks. And it was a big one for us to get working all the way through to these rooted plants here now. And it seems to be performing quite well in culture. So if I summarize our current position in culture at the moment for the different in-demand rootstocks the industry have told us they'd like, we've basically got commercial scale readiness for both reed and Velvic, with only a little bit more optimization needed for the rooting percentage in Velvic. Here you can see it's still quite difficult to root Velvic, but the acclimatization rate is really good. Zutano, again, needs a bit more rooting improvement, but great acclimatization success. Kid and Juicer are looking really good with their rooting percentages now. Kid's easy to acclimatize in our hands. Juicer, we're still improving the acclimatization. So we're hoping that basically starting from this, which took years, and this, which took years, these guys have taken not long at all, really, using our combined knowledge and information from working really hardly on these two. So every kind of new cultivar we get in now, we're finding it's much, much easier to start to optimize straight away and get things working. So these are just showing plants that have been acclimatized out of the culture system. So the real world is a really scary place for a tissue culture plant. Tissue culture plants are very spoilt, they're very precious. They've come from a beautiful environment where everything's given to them, and you can't just put them out in the sun, they just shrivel and die. So you've really got to slowly introduce them to sunlight and lower levels of humidity. And this on the left here, you can see a reed plant that's come out of culture in the nursery, and you can see it's flowering straight away. So we're putting mature tissues in, and they're retaining its maturity during the culture process, and it's mature when it's coming out. And here we have Velvic, Zutano, Kid, and Juicer also nicely acclimatized. So lab to nursery, this is where we've taken plants out to Duranbah, New South Wales, and got them out to the nursery ready for field planting. These are the different cultivars I've mentioned, or rootstocks. And Jani's also done a really good job of making sure she's field planted her plants and checked that their morphology is normal. So here you can see a really nice root system for reed. The fruit, as expected, flowering is normal. Everything's looking good. Same for Velvic. Velvic is 2 years old now. Great root systems. Terrible eating fruit, you don't want to eat this, but this is what the fruit looks like for this rootstock. So I'm going to change tack a little bit now and talk about the work that CQU did before I talk about our field trials, because I think this really feeds into our field trials actually. So these guys, led by Talitha Best, did a whole bunch of surveys for us to assess end user perceptions of tissue culture. So they did an international and domestic survey. And if we focus on the domestic respondents, we had 42% identifying as growers or farmers, 19% as breeders and researchers, 11% agronomists, 11% consultants, and 8% as nursery or supply chain. So there's a few major findings they found in their survey. The main ones at the start were, yes, more than 72% cannot access enough plants. More than 56% can't access the quality of plants that they want. And more than 53% can't access the cultivars they want. So this validated for us the problems are not just perceived by us, but they're actually real in the industry, which is really important, obviously, for us doing the research. The second survey finding that hit home for us was that 45% thought they already had the skills and knowledge needed to work with tissue cultured avocado plants, but 55% thought they didn't. So there's an education gap there, or barrier to uptaking these tissue culture plants at the end of the day. However, 2/3 of domestic respondents believe that tissue culture could be beneficial, which is good. The most desired characteristics they wanted was quality of the root system, disease tolerance and disease-free status of the plant, and of course, fruit quality. But the major thing they said that before they uptake any tissue culture plants, they want to know that they're performing well in the field. And they also want to hear professional recommendations from others they trust. So field testing performance is essential for industry acceptance. So this is Maddie here. Over the last 3 years, she's painstakingly wheelbarrowed many plants to field trials around Australia. This was to test seamless integration of our tissue culture trees in commercial orchard management strategies. So I'm going to briefly talk about those trials. There was various climatic conditions, and we were looking at performance of a tissue cultured rootstock grafted with a commercial variety on top relative to the traditional seedling rootstock as the comparison. She did large-scale transfer of our plants from culture to the nursery at Anderson Horticulture, showing beautiful root systems. They transplanted really well, and there was no difference in graft success of tissue culture plants versus seedling controls. This was all done using reed rootstock, which was the first to come out of our system. We then set up field trials around Australia with our grower partners. So Donovan Family Investments supported our work in Childers. We had 2 farmers, pioneering farmers up in far north Queensland in Lakeland, which are banana farmers that have turned to avocado. We then had 2 farmers also in Western Australia, some of the biggest orchards in Australia. So a wide range of climatic conditions. Maddie found there's no issues with establishment, so 90 to 100% establishment success, very low losses. And all in all, there was 240 tissue culture trees in these trials, alongside seedling controls. So I'm going to talk about the Childers trial today. This is our oldest trial. It's been in the ground 3 years now, with Hass grafted on top and a randomized design with limited clustering. So Maddie's been measuring, with the help of her amazing mum and myself, the height gain and diameter growth of plants over time. So here on the left, you see the height gain. And this is just basically showing you that seedling rootstock grafted plants and tissue culture rootstock grafted plants, they're growing well. Tissue culture is gaining height. It's growing just as well, if not better than seedling rootstocks. So they're performing well in the field in terms of that measurement. And over here, if you can see diameter, so growth of the diameter of the plant over time. Again, tissue culture slightly bigger gain in diameter growth, but not significantly different to seedling, but performing just as well in the field at 2 years. When we look at the fruit, so bearing in mind by year 2, this is not commercial scale of production yet, the plants are still quite young, so you're not going to be getting the huge amount of fruit that you'd see in a 5 to 7 year old tree. But you can see already no difference in tissue culture produced amount of fruit compared to the industry standard. The yield, no different, no statistically significant difference there. The mean fruit mass, so the size of the fruit, was slightly higher in year 2 for the tissue culture trees. And here's Maddie's mum, she's amazing, packed lunch and everything. And then year 3, so year 3 we just collected a few weeks ago up in Childers. Again, the tissue culture plants are yielding just the same as what you'd expect any old plant to do, seedling plant. No difference in yield or fruit count, and this time the mass is the same as the seedling as well. So basically, the trees are performing exactly how Maddie expected them to perform, just as we want them to perform as well, just like the industry standards, which is really busting that myth that the industry thinks they don't have the skills already to grow tissue culture plants. Well, they do, because you actually don't need to do anything different. Just put your plants in the ground, treat them as you would a normal seedling rootstock, and you'll be fine. I'm going to lead now into some financial analysis that our USQ partners did. So this kind of came out of a survey too, that money was perceived to be a barrier for using tissue culture trees. They thought the cost of trees was going to be a problem. So our team at USQ, headed by Professor Jeff Slaughter, are business and economic analysts. And I'm going to totally admit that this goes over my head, and I'm not this way inclined at all, but I'll try to regurgitate what they told me. So basically what they did is they modeled different scenarios of an orchard. So they started with data that they collected from grower visits, nursery visits, agronomists, academics and researchers, industry reports, and they set up basically their base field. So a planting density of 312 trees per hectare, 10% mortality, plant price, $35 a tree. This is a normal price for a plant in Australia. They modeled the business horizon over 20 years on a size of a farm of 25 hectares. They then looked at different financial matrices, gross margin, net present value, annuity, maximum negative cash exposure, payback period, internal rate of return, based on the fact that they're assuming an incubation stage of 0 to 2 years where you're not getting any yield, a first bearing stage of years 3, increasing yield over time, a required rate of return of 7%, and an orchard capital structure of 100% equity. So then what they did, I've taken a selection of the scenarios that they modeled. So scenario 1 is the traditional propagation method. And then they've modeled these possibilities that have come from tissue culture. So tissue culture allowing you to get your plants as you need them. So, 1 year earlier, I'm getting plants in the ground. Tissue culture allowing you to get those really high performing elite rootstocks, giving you a potential yield increase. Low on plant mortality was something they wanted to see, if that would have an impact on the grower profitability. And then they combined 1 year earlier yield with a higher plant price. So this was an interesting one to see if they doubled the plant price, how would that actually affect the grower's return over time? So what they found was, in the base scenario, you get about $19,000 a year cash flow per hectare, an internal rate of return 32%, and payback period of 6.5 years. If you look at earlier planting and yield increase, you're getting $3,000 more per hectare per year, and reducing your payback period by 2 years, and getting almost 13% internal rate of return increase. So trees in the ground earlier is highly profitable over the life of the orchard. Sorry, this meant earlier planting and therefore earlier yield of the plants. And then if we look at earlier yield, so the same model as the grey above, but then you've doubled the price of the plants that you're putting in the ground in the first place, it's not actually having much effect. You're still getting an extra 2,000 based on your base scenario per year, per hectare, and reducing your payback period and increasing your internal rate of return. So this was something that basically says over the life of an orchard, higher planting cost doesn't make much difference. It's really getting the plants sooner that's going to be a big thing for growers. So just busting that myth that tree price actually has limited influence over the life of the orchard. And another thing, we don't actually expect tissue culture trees to cost much higher. They're not commercially sold yet, but from what we estimate, we don't think it's definitely not going to be double the price. And we would estimate around the same as what you can get at the moment. So this is just some pictures of our growers' day that we held the other day. We had about 80 people come along, which was really awesome. Got some really good engagement from growers and industry to showcase what we've done. And in summary, avocado tissue culture is possible at a commercially relevant scale. We have 5 rootstocks ready or accelerating towards commercial readiness level. We've tested 240 trees for their performance up to year 3 so far across Australian conditions. Most growers agree that plant supply is a problem and are willing to uptake tissue culture plants. Education and engagement, it will be really important. The avocado market is still growing, and a major financial benefit at the grower level will be access to plants on demand. So I'd like to just draw your attention to a booklet that we've created as part of this project information pack, and that will be going up on the website, our website here pretty soon if you want to learn more or have a kind of take-home information on what our project has achieved so far. And of course, contact Nina or I at any time, or the team. And now we've got some smashing avocado tissue culture Instagrams, the first Instagram tissue culture avocados. And yep, what's happening next? We're basically gonna start now on macadamia. We were successful with an ARC linkage. We're gonna try the same innovations for our macadamias, which are also super, super important for Queensland. We've got some great partners on board for that, as well as all the work we're doing in multiple other aspects of avocado and other horticultural crops and native species. So watch this space. And yes, I'd love to thank everybody, all these guys, Johnny and Maddie, especially for all their hard work on this project, and Chris, and Nina, of course, our fearless leader, superhero, USQ, CQU, Advanced Queensland for their funding, and the Queensland government, and obviously all our grower partners that really supported our research. So thank you to everybody. Open for questions.
Fantastic. Thank you very much, Dr. Alice Hayward. Excellent talk, as usual. We do have a few questions on the Q&A, and this one is from Jacob Joes, and he asks, does the age of the mother tree from which the meristems are harvested for tissue culture have an effect on the tissue culture success percentage and quality of the trees produced?
Oh, that's a really good question. At the moment, we're using mature tissue. So we can guarantee that our system uses mature tissue that's taken from budwood from field-grown trees. We didn't want to use anything that wasn't mature and productive because we want to make sure that it's really translatable to industry for cloning those rootstocks that have been selected in the field. So we haven't done a big test of how easy the system is across seedling versus mature plants. Seedling plants are notoriously easier in tissue culture, which made mature a much bigger achievement for us. So everything we use has come from field-grown mature tissue.
We've got a couple more questions that have come through. This one's from an anonymous. Do you know if tissue culture produced adult trees have comparable root architecture and root biomass as the seedlings grown adult trees? And do you think tissue culture adult trees would have as good anchorage as seedling produced trees to be able to withstand wind?
Yeah, I think this is something we'd really love to be able to quantify better. Just from our observations, obviously, and our grower observations and nurseryman observations, they think the root systems look incredible. They're very complex, vigorous, and they establish really well, obviously. And so far, we haven't had any issues in the field. But I think it would be nice to be able to quantify the root systems better. And it's something that would, and also show up over time as these plants are in the field for longer. But they really are beautiful root systems. It's not like a traditionally clonally propagated plant where you have to go through that massive process in the nursery. Tissue culture root systems are known for being quite vigorous and really sturdy and hardy. So I don't envisage it's going to have those same problems that some people find with the clonally produced nursery plants.
Certainly round out the complete story, that's for sure. We've got a couple more. Will increasing supply of the plants affect the fruit price?
Yeah, this one comes up all the time.
Yes, it does.
Yeah.
We've had some discussions ourselves on this.
Yeah, I had a slide on this, but then I should have left it in. So the USQ guys did a lot of market analysis and financial modeling of the existing market, and basically what would happen if you flooded it. At the moment, they've shown historically there's no relationship over a whole year between fruit price and supply and demand. So at the moment, the demand is growing and the industry's growing enough that they haven't seen any of that effect, and they can't see it they haven't foreseen it happening anytime in the near future. And I guess, you know, as things always do, I guess if you're producing way too many than what people want, eventually it will get to that point. But I think that's where a lot of the work on trying to boost export markets and things like that would be important as well.
People get to eat more avos, I suppose. So they're still in that demand. We've got two more questions. And will a grower be, for this is from Katia, will a grower be able to select his or her favorite tree and send a sample to get multiplied?
Oh, that would be great.
For a price, yes.
Yeah, we'd love to talk to people interested in that, but we're a lab, so we're doing the research. We're not the commercial producer of plants, so that we are licensing the technology to other companies that will produce the plants in tissue culture. So I think that's something that could be explored in the future.
And we've got one from Milos. And if the majority of the growers took this on board, how many ready-to-plant plants would you need to produce yearly?
Oh, what is it? Anderson Horticulture would know. Yeah, Anderson Horticulture would know this based on the demand currently is. A few 100,000 a year, I thought, was what their demand is. But there's more growers coming on, and there's also the yearly cycle of plants needed. It's a good question. I'm not sure exactly the number.
Lots.
Yeah.
Definitely. All right. So last question now. Do you think that establishing tissue culture in avocados will encourage greater real-world genetic diversity across global orchards?
Do you mean by accessing more genetically diverse rootstocks and getting them out to Australian growers?
Yeah, let's try that.
Yeah. Or it could be in relation to this seedling versus clonal debate, I guess.
Yeah.
So I think the point is with tissue culture, everything's clonal, but where you're actually getting the accessions or the elite breeds that people are trying to develop that are overcoming a lot of the problems. So if it's salinity or disease resistance that you may not get in your seedling rootstocks. So it allows access to actually a different variety of rootstocks to choose from. that you'll be able to get much quicker. So that way you can basically get a selection of what you want, as opposed to seedlings from just one or a few rootstocks, if that makes sense.
Definitely does. We've got one last question. And when going commercial, how many trees would be able to be produced from one single tree? And i.e., will we find hectares of planted clones in orchards?
Well, it will depend on the rootstock. So they all behave differently in culture with different multiplication rates. If you think about one single bud off a reed tree, you can get 500 plants from that. So on a whole tree, if you were to take every single bud off, you could make millions, I'm sure, from a whole orchard of plants. Just depends on the supply of the tissue culture company and the demand. But it's kind of a kind of a conveyor belt system where the demand can actually be scaled, or the supply can be scaled based on what the demand is as well. So you always have the cultures sitting in tissue culture. So from the point at which you order a plant, you're not starting from scratch of getting the bud into culture. You've already got cultures there ready to be rooted based on your demand. Yeah.
Oh, well, that's it for the questions, Alice. I'd like to thank you for an absolutely fascinating and fantastic talk, as usual. Just a reminder that we do have these seminars every Tuesday, and the next one is next Tuesday on the 22nd of June, and it's titled Untangling Genotype by Management Interactions in Multi-Environment On-Farm Agronomic Experimentation. And this is presented by Professor Daniel Rodriguez, and it's a bit of a mouthful of a title, but it will be an excellent talk as well. Professor Rodriguez is also from QAFI, Centre for Crop Sciences, and you can always look at the QAFI Science Seminars webpage for more information. But I'd just like to thank everybody for attending our science seminar today. The more attendees, the better it gets, definitely. If you enjoyed today's seminar, please do the poll at the end. And if you haven't already signed up for our seminar invitation list, please do so at all the W's, QAFI at quafi.uq.edu.au/science_seminars. I'll say thanks again, and thanks to Alice, and we'll see you on the next one.
Thanks, Carl. Thanks, everybody.
Thanks, guys. Cheers.